my subject for this week is looking at more complicated repeated measures designs. So this goes back to some stuff that we saw a couple weeks back in Lab 5, where we looked at a simple repeated measures design where one variable was repeated over several time points. So this is technically a type of MANOVA, although it's often just called a repeated measures ANOVA. And we're going to go and look at some more complicated versions of repeated measures ANOVA now. So when we have repeated measures analysis, we have two potential types of variables, within subject and between subject. And just as a sort of aside, there's another popular statistical method that is instructed at Fordham as well. There's a class about a whole class about this method for handling variables that might vary either within or between subject. And this is actually a class that I taught last semester, which is called Hierarchical Linear Modeling. And so this is a very popular method as well for handling repeated measures. It can do some other things too. It can do some things that repeated measures ANOVA can't do. Repeated measures ANOVA does some things that HLM can't do, but there's a lot of overlap between them. They can do a lot of the same things. And there are some differences. So some of the differences, for example, are in terminology. So in repeated measures ANOVA, we tend to differentiate between within subject and between subject variables, what we call these variables level 1 and level 2 in HLM. And so these are the same idea. One of the practical differences is the structure of the data. So in repeated measures ANOVA, we tend to have wide data where we have one row per subject and several columns for each repeated measure. Whereas in HLM, we have long data where we have multiple rows per participant with some kind of identifier variable that shows to which participant they belong. These procedures use different algorithms. So repeated measures is based on ANOVA. That's the actual statistical procedure. While as HLM is typically based on some sort of maximum likelihood algorithm. There's other possibilities too. Sometimes you'll see Bayesian HLM is typically done with Markov chain Monte Carlo, MCMC, but the most common flavors of HLM are done with these maximum likelihood algorithms. The conceptual framework is different. So again, the analysis of variance ANOVA is the framework for repeated measures, while HLM is more from the regression sort of lineage. They both have some advantages. So repeated measures ANOVA is quite a bit simpler computationally. It's easier on your computer generally. You'll usually be faster. HLM will sometimes take a little, usually not too long, but sometimes it'll take a few seconds to fit a model. Repeated measures ANOVA can handle certain cases that HLM can't. So it can handle these cases where there's negative within subject correlations, which are very atypical, but they are possible. And so this is something where you need to use ANOVA, you can't use HLM. ANOVA is a little bit more well established. It's been around quite a bit longer. It's a little bit better known probably across disciplines, whereas HLM is a little bit more of the new kid on the block. And in repeated measure ANOVA, you can include multiple dependent variables over time. So you can look at several different dependent variables. We'll look at an example of this today. On the other hand, HLM has more intuitive parameter interpretations. You don't need to do lots of different steps to sort of interpret your results. You can directly estimate what are called fixed and random effects. So fixed effects would be like population level effects. So these are parameters that apply to the population at large. And then random effects are specific to the individual subject. So random effects would be like effects that are specific to the specific participants. HLM is much easier to code, in my opinion, especially in R, and has excellent support in R with the LME4 package and some others, but mainly LME4. It's a wonderful package. In some ways, I feel like it should almost be included in base R at this point, but it's one of those top tier packages. So HLM can also more easily accommodate certain types of unbalanced designs where you don't have the same, not all participants are measured at the same time, or you have different numbers of measurements of different participants. So in repeated measures ANOVA, you'll typically see each participant is measured at the same time points, the same numbers of times, and anything where that's not the case is considered missing data. HLM is more flexible. It can handle cases where participants are measured at different times. It can handle cases where you don't have the same numbers of measures per participant much more smoothly than repeated measures ANOVA. And they both also have some disadvantages, which we mentioned a little bit already. So repeated measures ANOVA, we'd have to sort of go piece by piece and interpret results as we go. So we start with these very general omnibus tests and get more specific as we go. And eventually we drill into the results of interest. Our support for repeated measures ANOVA is a little clunky. It's not my favorite. It could almost certainly be better. I think it's partly because HLM is so popular, and R really never got off the ground with repeated measures ANOVA. In a lot of cases, HLM has supplanted repeated measures ANOVA for a lot of the most common things that you'll see, but again, not everything. So it's important to know both. Repeated measures ANOVAs can't handle these unbalanced designs as well, or cases where there's missing data. And on the other hand, HLM can't handle these cases with negatively correlated within subject variables. Again, these are very unusual, but they can happen. And this is just not something that, this is a violation of HLM assumptions. It's not something HLM can handle at all. HLM, because of the maximum likelihood algorithms, they can fail to converge. So they can run into problems where they'll just tell you they couldn't find a solution. And you either have to find a way to reconfigure your model, or you may not be able to fit your model at all. So HLM can also only handle one dependent variable at a time generally. So it's not multivariate in the sense that we've been discussing so far. It is very multivariate in other ways, but not in terms of having multiple dependent variables. And one thing they both have in common is that the framework for effect size is still a work in progress. Just like repeated measure ANOVA and HLM, there's ongoing discussions about what the best way to think about effect size is. And this is something that's still sort of up in the air. So that's just a sort of aside about HLM. If you haven't taken it, I highly recommend that you take the HLM course. I know it's going to be required for anyone who hasn't taken it who's in PQP. But if you're not in PQP and you're looking for another stats class, it's a really, really useful class, really great class. Anyway, more on repeated measures ANOVA. So in repeated measures ANOVA, each within subject variable is considered a dependent variable. Then we use our MANOVA methods to allow them to covary. So in the first time we looked at repeated measures analysis, we looked at how a single variable can be measured at different time points. And so this would be a one-way repeated measures design. So today we're going to look at two-way within subject repeated measures, where multiple variables are measured simultaneously at different time points. And then we can also look at how we can mix within and between subject variables. These are called mixed models in the ANOVA terminology. And we'll see some examples of this as well. So we're going to look again at simulated data. I've been liking this idea of using simulated data where I can sort of tell the story that I want to tell using data. So here we're going to imagine that we're a data scientist working at Spotify. And this is sort of based on some stories a friend of mine who worked at Spotify has told me. I sort of tried to simulate some of the trials and tribulations he goes through. So your task is to study new user engagement for both music and podcasts over time. And so you start by tracking weekly engagement levels on both dimensions over a period of 3 weeks. So your data has 6 columns. Anything with an M prefix is music, and anything with a P prefix is podcasts. The suffixes T1, T2, T3 represent weekly engagement levels, and we want to find out how people's engagement levels change over time, and if the changes are different for music and for podcasts. So here's what our data looks like. You see we have 6 columns. 3 M columns at 3 different time points, and 3 P columns at 3 different time points. So like our original repeated measures example, we need to set up our iData and iDesign. And what these stand for is intra-subject data and intra-subject design. So this is how we set up the within subjects piece of our model. So before, these were simply the levels of our single one-way factor, but now we have 2 factors, time and media. So the media are music or podcasts. So the way we're going to set up our iData is we're going to have a data frame with 2 columns. They're both going to be factors, and the 2 columns represent all possible combinations of the prefix or suffix. So they're going to really represent our column names in our actual data, but they're going to be separated into the prefixes and the suffix. So both prefix and suffix should always start with a letter character, not a number. So if this isn't the way your data is named, you need to change the names of your columns in your actual data. That way the iData will work. So using a number or the wrong type of character will trip things up. We also want to set our contrasts to effects coding because dummy coding won't work with the estimation of this model in R because R is weird. So set my contrast, and then I'm going to build my iData. So you see, I'm just creating one factor called media, that's just MMM, then PPP, and then another called time, which is T1, T2, T3, T1, T2, T3. So we see that this is just a representation of our column names, and they're just separated out by prefix and suffix. And so we want to double check that our iData is set up the right way. To do this, we can use the paste zero function to check that the columns of the iData match, the columns of the iData reproduce the column names of our actual data. So here I'm going to use paste zero to paste together the two columns of my iData. You see those give me back the same column names from my actual data, and these should be in the same order as well. And I can check this with the all equal function to see if these two match up. And so we see that the pasted elements of my iData match up to my column names in my actual data. So now I can run the ANOVA, and like we saw before, I first fit an LM object with all my variables as dependent variables regressed on an intercept. So just regressed on a 1. So next I enter this into my ANOVA function from the car package, that's the big A ANOVA, with my iData as the iData I set up. And then for the iDesign, I do tilde on the interaction between the column names for my iData. In this case, I name them media and time. And so here is my ANOVA table. This is for my omnibus tests. Notice that I'm calling type equals 3 for type 3 statistics, and then getting the Wilks for my test statistic. The test doesn't matter that much, but we've been talking about lambda, so let's stick with that. So our interpretation here is that the interaction is significant. Just like in the 2A between subjects we saw earlier, we want to look at cross contrast. And the other way to interpret this significant contrast is that what it's saying is that the change in engagement over time, or the difference at the difference in engagement at the different time points depends on the type of media. So it's not the same for music and podcasts. I poked around and figured out some more ways to test the crossed, or just the general contrasts with the linear hypothesis function. And so this would also work with the one-way within subjects design we saw. And so the idea is we have to set the hypothesis matrix equal to 1. So this represents the intercept we regressed on. So the hypothesis matrix is talking about the between subjects factors. But then we can use the P equals argument for the within subjects factors. But this has to remember, this has to be a matrix object. It won't work if it's just a one-dimensional array. So it has to actually, you have to call it as a matrix object. So here, what I'm doing is I'm testing the first two columns of my data against one another. So I'm comparing music at time 1 to music at time 2. And here's the full output when we see that this is significant. And so here is podcast at time 1 to podcast at time 2. So this is the same order as the columns in my data. It just shows me back this response transformation matrix here. And so this we see is not significant. So we see there was a significant difference between time 1 and time 2 for music, but not for podcasts. So engagement in music was significantly different at times 1 and 2. However, engagement in podcasts was not significantly different at times 1 and 2. And so I'm just going to quickly check my data here to check the mean to see if I can sort of get a sense of what's going on. There are much more in-depth ways we can probe this, but this is just a simple way. So I'm looking at the means. And we see that it looks like music is increasing over time while podcast is staying relatively flat over time. One better thing we could do would be to plot this. We could plot this with confidence intervals, and we did see how to do these confidence intervals back in Mod 5, and that will be part of the extra credit for the assignment. So we've sort of figured out that we need to intervene and boost podcast engagement. It seems like music engagement ticks up as people use Spotify more, but we're not seeing an increase in podcast engagement. So our user researchers, who are another team of people working on this stuff, design a treatment and ask us to run an A/B test. The A/B test is just a weird term that data scientists and tech people tend to use for an experiment. It's the same thing. I'm not sure why they use a different terminology, but this is just essentially an experiment. So we're going to set up a test where we randomly assign 250 new users to the control condition and then 250 to a treatment and see how that affects podcast engagement. So we're not looking at music engagement here. We're just looking at podcast engagement. We could continue to look at music engagement, but that would start getting a little complicated with the design. So we're going to keep it a little simple and just look at podcasts. So here's a slightly different data set. So where we split the participants into conditions A and B. And so we see here podcast engagement across 3 time points in the different conditions. And so we have only one within subjects factor now, which is podcast engagement. But we do have a between subjects factor, which is the condition. So the between subjects factor is going to go on the right side of the model, replacing the one we've been regressing on as the intercept. So within subjects go on the left side, between subjects go on the right side. For my I data now, I only have one factor that I have to put into a data frame, just the podcasts. So I don't need to separate this out by prefix and suffix. That's only for when we have 2 cross factors. And then so I'm going to call my ANOVA and get my results. So here we see that the interaction is significant. So this tells us that, that there is a difference in the 2 experimental conditions in podcast engagement over time. So this is a sort of hint that our intervention helped. And so now I need to merge what we learned about between subjects ANOVA and within subjects ANOVA to do a little bit of both. So I can go back to the suppress the intercept method that I used last week for the hypothesis matrix. This simplifies what we can do there, because then we can just treat each regression coefficient as the mean of one of our within subjects factors. The other option is not to do this and then just make sure that we're treating it in the regression framework that we talked about in the last slide deck. But because we only have one within subjects factor, suppressing the intercept does simplify things. So then I can also use P to specify the within group contrast. So first we're going to compare time 1 to time 2 in condition A, and then we're going to do the same thing in condition B. So here I'm specifying, since I suppressed the intercept, my first regression coefficient is the mean of group A. And then I'm going to look at, in the P argument, the 2 time points I want to compare, so 1 to 2. So this result compares time 1 to time 2 in condition A. We see this is not significant, so this is the same result that we saw before, because this was comparing podcasts time 1 to time 2 with no intervention. This should be the same result that we saw before we started in our first set of results. And so now we're going to look in the condition B. That should say condition B, not condition A. And so we're going to switch it over to condition B. And we see this is significant. So it does seem like our treatment was successful. What we know now is that there was a difference. What the test tells us is that there was a significant difference between time point 1 and 2 in the treatment condition. But again, we can use other ways to probe our data. We could do, a univariate test in condition B, or univariate ANOVA for the different time points, or we could just do a one-way multivariate test, one-way one-way repeated measures ANOVA just in condition B and do some pairwise contrasts. But we could also just kind of take a look here and we see that it does look like engagement is increasing over time. And again, there are definitely more in-depth ways we can probe this, but I can only throw so much at you in one class. So this is in the right direction. This is what we wanted to see, that the treatment was boosting engagement. So podcast engagement improved in the treatment condition. Here are my statistical results.